Probability is a mathematical concept that quantifies the odds of the occurrence of a random event. Probability is expressed through numbers between 0 and 1, meaning that if an event has a probability value of 0, it is impossible for it to occur, and if the probability value is 1, it is certain to occur. Relative probability is calculated by dividing the number of favorable outcomes by the total number of possible outcomes. For instance, if in this situation, the favorable outcome is rolling out a green marble from this bingo machine, the number of favorable outcomes would be 1, as there is only one green marble in this cluster. There are 14 marbles in this machine, including the green one, so the total number of possible outcomes would be 14. There are a total of 14 marbles that could possibly be rolled out. In conclusion, the favorable outcome, 1, divided by the total number of possible outcomes, 14, is 1 over 14. The number can also be expressed as a percentage, in which case the probability would be 7.143%, and it can also be expressed as a decimal, in which case it would be 0.071. Probability is a very useful tool which is applied to many real-world situations, including weather forecasting, insurance, stock market predictions, traffic signals, and most importantly, genetic diseases.
Genetic diseases are caused by genetic mutations, and genetic mutations are caused by structural changes in the building blocks of DNA. Mutations can be considered as a variation in the genetic material of an organism. They may be caused by disorders in a single gene called monogenic disorders or multifactorial genetic disorders, which are caused by interaction of multiple genes with the environment. Mutations can be caused by a number of factors. Causes include chromosomal damage, such as chromosomal breakage, which leads to the loss or dislocation of genetic material, and chromosomal duplication, which refers to the unnecessary replication of chromosomes. The cells then follow the discrete instructions of the DNA, often causing abnormalities.
Genetic mutations, and therefore genetic diseases, can be passed down through multiple generations. The ability to pass on inherited features to the next generation is encoded in DNA. Some organisms, like python snakes and starfish, can reproduce asexually. Some methods of asexual reproduction include binary fission, where a parent cell divides, and budding, where the offspring develops from an anatomical point of the parent. In such asexual reproduction, a single organism single-handedly reproduces their own offspring. That offspring then inherits all of their DNA from the single parent. However, in organisms that receive their DNA from two parents, one copy of DNA is inherited from their mother via the egg, and the other is inherited from their father through the sperm. A sperm and an egg each contain one set of 23 chromosomes. After the sperm fertilizes the egg, 2 copies of the chromosomes of each parent, and therefore 2 copies of each gene, are present in the egg, for a total of 46 chromosomes in the offspring. As the parent's genes are passed down to their offspring, the genetic disease is as well. Each genetic trait in a genotype is represented by alleles, which are assigned the label of dominant or recessive. Dominant traits are written in capital letters, and recessive traits are written in lowercase letters. An offspring inherits a copy of each of their parents' genetic traits. Dominant alleles represent traits that are much more likely to show up in the physical appearance or phenotype of the offspring, compared to recessive traits, which may be carried by the child but not expressed in their phenotype.
In a recessive trait, both parents must carry the gene for it to show up in the phenotype of the offspring.
The general principles of inheritance present in many species in nature follow Mendel's laws of inheritance, the law of segregation, and the law of independent assortment. However, Mendel's laws may not accurately convey the intricacy of genetic relationships in all circumstances. The law of segregation states that each parent passes down their allele to their offspring at random, and the law of independent assortment states that alleles are inherited separately, meaning the allele inherited in one gene does not impact the inheritance of another. There are exceptions to Mendel's law of segregation and independent assortment, such as genetic linkage and epistasis. In genetic linkage, the closer 2 genes are to each other on a chromosome, the more likely it is that they'll get inherited together. For example, the gene for eye color and hair color sit closely to each other on a chromosome, as they both relate to pigment and color. This is why blue eyes and blonde hair, or brown eyes and brown hair, are likely to be inherited together. Epistasis is an occurrence where a gene is stopped from showing up in the phenotype as it is masked by another gene. Epistasis can be observed in Labrador retrievers. Labrador retrievers' coat-color genes are actually only black and brown. However, it is very common to see light-coated Labrador retrievers, and this is because extension genes, which are recessive epistatic genes, prevent the black and brown pigment from showing up on the fur. Genetic linkage and epistasis prove that genes can in fact interact with each other, unlike what Mendel's law of inheritance states.
The probability that an offspring will inherit a genetic disorder can be calculated through methods such as the Punnett square. They help predict the variations in genotype that can become from crossbreeding two different organisms. So, the Punnett square can be used to determine the potential genotypes of the offspring of a particular couple.
For instance, Punnett squares can be used for monohybrid and dihybrid crosses. The mono and monohybrid refers to the fact that one single gene is observed, while hybrid refers to the mixing of two different types of alleles. On the other hand, the di and dihybrid refers to the fact that two genes are observed, and hybrid refers to the mixing of two types of alleles as well. To illustrate, a monohybrid cross would examine hair color, which is a singular trait. In this example, 100% of the offsprings will have black hair, but will carry the recessive gene for blonde hair, while a dihybrid cross would examine hair color and hair texture, which are two different traits. In this example, the number of squares in which the phenotype is black and curly hair is 9 out of 16, the number of squares where the phenotype is black and straight hair is 3 out of 16, the number of squares where the phenotype is blonde and curly hair is 3 out of 16, and the number of squares where the phenotype is blonde and straight hair is 1 out of 16. This 9:3 to 3:1 ratio is Gregor-Mendel's classic ratio for dihybrid crosses. Most dihybrid crosses will have this ratio. However, in a dihybrid cross, there are a few assumptions being made relating back to the Mendel's laws of inheritance. For one, it is assumed that, as the law of independent assortment states, each of the genetic traits are inherited separately and do not interact with each other. However, the law does not apply to all situations. For example, in a dihybrid cross for eye color and hair color, factors such as genetic linkage and epistasis, discussed earlier, could skew the results. Although methods of calculating probability in the field of genetics, such as the Punnett square, can be incredibly useful, they come with their limitations as well. For example, calculated probability using a Punnett square is a big simplification of the complex processes concerning genetics, and may not take into consideration the impact of various other genes. Additionally, using probability in the science of heredity through methods like the Punnett square does not account for the impact of environmental factors. Such environmental factors include diet, physical activity levels, climate, oxygen levels, and presence of mutagens. Examples of mutagens include X-rays, ultraviolet rays, and alkalizing agents like ethyl methane sulfonate and N-methyl and nitrosurea.
That was an interesting video that I just watched. Oh, I'm having a heart attack.
Sir, are you alright? Familial hypercholesterolemia. It runs in my family. What is familial hypercholesterolemia?
Familial hypercholesterolemia, also known as FH, is a genetic disorder that affects 1 in 250 people and increases the risk of coronary heart disease at a young age. Birth with high levels of low-density lipoprotein, LDL, cholesterol can lead to heart attacks in children. Cholesterol is a fat-like molecule found in the cells of the body. When there is too much cholesterol in the blood, it deposits on the walls of the arteries, increasing the risk of heart disease. As the cholesterol collects on the walls, the arteries become narrow and the blood flow to the heart is decreased.
FH patients have high amounts of low-density lipoprotein LDL cholesterol. LDL cholesterol is commonly referred to as bad cholesterol. People with familial hypercholesterolemia have high LDL cholesterol levels because they're unable to appropriately eliminate LDL from their blood. The liver is the organ in charge of LDL clearance. High LDL cholesterol levels in the blood can raise the risk of a heart disease or a heart attack.
The gene mutation that causes familial hypercholesterolemia is located on chromosome 19. It encodes information for a protein known as the LDL receptor, which is in charge of removing LDL from the blood. 1 in every 500 people possesses the mutant gene responsible for familial hypercholesterolemia.
The gene mutation in the LDL receptor causes the LDL to build up in the arteries. As mentioned, LDL blockage in the arteries causes the heart attacks that come with FH. This Punnett square expresses the genotype of the offspring of one parent with FH. The father carries a heterozygous dominant allele for FH, and the mother does not carry the gene at all. She carries 2 recessive alleles. 2 out of the 4 squares have a dominant allele, meaning there is a 2/4, 0.5, or 50% probability that the child will inherit FH. The 2 squares are the measured favorable outcome, and there are 4 squares in total. So, the probability would be equal to 2 divided by 4. But
Should the risk of your child inheriting a significant disorder be a factor for couples deciding whether to have children?
People with familial hypercholesterolemia should definitely factor in their disease into their decision of having children. There are many ethical implications of having children as an FH patient. Having a biological child as a person with familial hypercholesterolemia means running the risk of the child inheriting the disorder, along with all the health risks, symptoms, financial, and social burden. Having the disease causes an increased likelihood of coronary heart disease at a young age. In the United States, coronary heart disease is the leading cause of death. The financial cost of FH over the course of 11 years was studied to be around $17,000 to $11,000, even without cardiovascular events. However, in the first year after a cardiovascular event, it was estimated that the patient had to spend upwards of $17,000 to $65,000 in treatment. The study came from the United States in 2019. Symptoms of FH include lumps around the eyes, knees, knuckles, or elbows, swollen or painful Achilles tendon, and yellowish areas around the eyes. Such financial burdens, physical symptoms can cause great social implications on the child, such as social isolation. The social, physical, financial effects could lead to mental health disorders such as depression and anxiety. So, patients with familial hypercholesterolemia should definitely factor in their disease into their decision of having children, and should preferably choose other ways of raising children, such as adopting instead of carrying biological children. Or they should consider not raising children at all.